Welcome to Part 3 of Chapter 3, The Cells and Tissues. In this video voiceover lecture, I'll discuss Section 7 on protein synthesis and Section 8, the tissues. Section 7, protein synthesis. Let's begin by talking about one of the most fundamental ideas in biology, the central dogma. It was first explained by Francis Crick of the double helix fame. The central dogma describes the flow of information that allows genes to be expressed. It begins with the exchange of information that allows DNA to be copied. You know now that copying our DNA is crucial for cell division. This process, where DNA is used as a template to make more copies of the same DNA, is called DNA replication. DNA can also be used to make RNA. This is known as transcription. In the last step, RNA is used to make protein. This is called translation. Remember that DNA and RNA are composed of nucleotides. In this way, they are written in the same language. When you copy the words from a speech or a book in the same language, it is called transcription. The term translation comes from the same term that is used to describe when you write something down in a different language. The two languages in this case are the nucleotides from RNA are used to write the language of protein in amino acids. Now's a good time to discuss the differences between DNA and RNA. Remember, DNA and RNA are both made out of nucleotides. And nucleotides are composed of three parts: a sugar, a phosphate, and a base. The sugar that's found in DNA is known as deoxyribose, whereas the sugar found in RNA is ribose. One of the bases that's found in the two molecules is also different. In DNA, we find the base thymine, which is replaced with the base uracil in RNA. The number of strands that are found in DNA and RNA is different. DNA we know is double-stranded, whereas RNA is usually single-stranded. Finally, the overall shape of the molecules is different. DNA has that nice, orderly double helix shape, whereas RNA is like a string that you've taken and balled up. This is called a globular or globular shape. In the two pictures below, you can see the different structures of DNA on the left and RNA on the right. There are three types of RNA that you need to know. The first one is mRNA. The M stands for messenger. This is the RNA that gets translated. This type of RNA gets read by the ribosome. Remember, ribosomes are molecular machines that synthesize proteins. How do they know how to synthesize a protein? This is because they read the message that's encoded by mRNA. The next type of RNA is tRNA. The T stands for transfer. So remember, ribosomes are synthesizing proteins. Proteins are composed of amino acids. So in other words, the monomer of a protein is an amino acid. There are 20 different amino acids, and proteins can be hundreds or thousands of amino acids long. It's the tRNA, or transfer RNA, that carries these individual amino acids to the ribosome. Finally, we have rRNA. The R stands for ribosomal. And this is because ribosomes are made in part of rRNA. Ribosomes are also partly made of protein as well. Transcription is mainly the process of RNA synthesis. In other words, making RNA. Remember, our DNA is found in the nucleus of our cells. The process of transcription requires DNA to make RNA, so transcription must be carried out in the nucleus. DNA is used as a template to make RNA. The enzyme that makes the new RNA is called RNA polymerase. The way this works is DNA is unzipped, like we described during replication. The bases, or the letters, A's, T's, G's, and C's, are exposed one at a time. A complement of the exposed DNA letters is added, one by one, to a growing RNA strand. So when a G is exposed on the DNA, a C is added to the RNA. When a C is exposed on the DNA, a G is added to the RNA. When there is a T on DNA, an A is added to the RNA. Finally, when an A is exposed on the DNA, a U is added to RNA, not a T. There are no T's in RNA. On the test, you'll be asked a series of questions about RNA synthesis. Particularly, you'll be given a DNA sequence, and you'll be asked to identify the RNA sequence that is made based on that DNA sequence. So, for example, what RNA is made from the following DNA sequence? If the DNA sequence is GATCGGAAT. Now let's discuss the last part of the central dogma of biology, or translation. To understand translation, you need to understand the genetic code. Genetic code is composed of codons. A codon is a sequence of 3 consecutive nucleotides, or bases, on an mRNA that codes for a single amino acid in a protein. So 3 letters on an mRNA code for 1 amino acid on a protein. This table shows the genetic code of life. All living organisms share the same genetic code. What this shows is a codon and what amino acid will be added to the growing protein from that codon. So during translation, you have a ribosome, and a ribosome is reading the mRNA. mRNA is a series of letters, A's, U's, G's, and C's. The ribosome reads those letters. If the first 3 letters that the ribosome reads is U, which happens to be the first codon ever discovered, then the ribosome would add phenylalanine as the first amino acid to that new protein. Let's say the second codon on the mRNA was CCC. Then the second amino acid that the ribosome would add would be proline. Let's say that the third codon is AAG. AAG is found here on the table, and that codes for the amino acid lysine. So the third amino acid in the growing protein would be lysine. And this would go on for hundreds or thousands of codons for individual proteins. When that one protein is done being made, there would be a stop codon that would occur. The 3 stop codons are listed here on this table. You don't really need to know the details of the special codons, the stops, and the starts. What you do need to know is how to read this genetic code table. So if I give you a codon, let's say AAA, you should be able to tell me on the test that that codes for the amino acid lysine. If I gave you the amino acid serine, you should be able to tell me the reverse, and that's that the codon coded for serine is either AGU or AGC. To clarify, translation is all about protein synthesis. During protein synthesis, the ribosome reads the message found on mRNA. The ribosome reads the mRNA 1 codon at a time. That's 3 letters at a time. Based on the codons, the ribosome will create a protein. The protein is made 1 amino acid at a time based on those codons from the mRNA. For example, the letters or the codon UUU will encode the amino acid phenylalanine on the new protein. Section 8, the tissues. A tissue is a combination of different cell types and extracellular matrix that function and work together. The word tissue in English comes from the French word for woven. The study of tissues is called histology, and this comes from the Greek histos, meaning web. There are 4 different types or categories of tissues. These are the epithelial tissue, connective tissue, muscular tissue, and nervous tissue. These different tissue types come together to form organs. Some organs, like the heart, contain all 4 types of tissue. So your heart is surrounded by epithelial tissue. The inside of the heart is also lined with epithelial tissue. There is connective tissue holding the heart together. The heart is mostly muscle, so it's mainly composed of muscle tissue. And there are nerves that innervate the heart to help control its beat. Let's begin this section by describing epithelial tissue. The structure of epithelial tissue is that it is found in flat sheets. The cells in epithelial tissue are closely adhered. That means they're squished close together. This picture below shows 2 different types of tissue: epithelial tissue on the left and connective tissue on the right. You can see on the left, the epithelial tissue cells are found very closely packed. The cells kind of look like the pulp in an orange. The purple dots inside the cells are the nuclei. On the right, we have connective tissue. Now, the connective tissue cells are actually widely spaced. They are these darker purple circles. Those are individual cells. And all of the stuff in between is considered extracellular matrix. So connective tissue has widely spaced cells. Epithelial tissue has very tightly, closely packed cells. Epithelial cells have something called a polarity. That means they have 2 different sides. The upper surface is known as the apical surface. And this is the part of the cell that comes in contact with the environment, like the air or water that surrounds our bodies or is inside of our gastrointestinal tract, or the lumen of a body part, so the cavity in your heart or the cavity in your blood vessels. The lower surface of the cells is called the basal surface. This is the side of the cell that faces the basement membrane, or is perhaps connected to the basement membrane. The basement membrane is extracellular matrix and connective tissue and helps hold our epithelial tissues in place. This diagram shows an epithelial tissue composed of a single layer of cells. These pinkish cells that are long and tall are epithelial cells. The surface that points towards the lumen or towards the air surrounding our bodies is the apical surface of the cells. Down below we have the basement membrane. This is connective tissue that's holding that layer in place. The side or surface of the cells that comes in contact with the basement membrane is the basal surface. Epithelial cells come in 4 different shapes. These are squamous or flat, cuboidal cube-shaped, columnar long and tall, and transitional. The shape can change. In this diagram, you can see the 4 different cell shapes of epithelial tissue. Here we have squamous epithelial cells. Notice that they are flat. Next to that, we have cuboidal cells, or cube-shaped epithelial cells. Here are the columnar cells to the far right. Again, they are long and tall. And then at the far left, we have transitional epithelial. These cells can change shape. They usually vary between squamous and cuboidal, depending on how stretched the tissue is. Transitional epithelium is mainly found in the urinary tract, in our urinary bladder and ureters. The structure of epithelial tissue can also be described by the number of layers. This includes simple epithelial, pseudostratified, and stratified. This diagram shows the three different arrangements or layers of epithelial tissue. To the far left is simple epithelial tissue. You can see the blue represents the basement membrane, and attached to that is a single layer of epithelial cells. Again, that's simple epithelial tissue. To the far right is stratified epithelial tissue. Again, in blue is the basement membrane. And above that is a stack or several layers of epithelial cells. In the middle is pseudostratified epithelial. It's technically a single layer of cells like simple epithelial, but under a microscope it appears to be stratified. And that's because the nuclei are scattered almost randomly like you see in stratified epithelial. And also some of the cells which are connected to the basement membrane don't make it all the way to the apical surface. And so under a microscope it appears to be several layers of cells, even though it's truly only one. Again, this is known as pseudostratified epithelial tissue. Let's move on to our second type of tissue, connective tissue. Let's first talk about the properties of connective tissue. Connective tissue is kind of like the garbage bag grouping of tissues. It's the tissues that don't fit into the other three categories. So if it's not muscular, nervous, or epithelial tissue, then it's probably connective. So some functions of connective tissue includes that it connects other tissues together. Connective tissue includes the ligaments and tendons. Connective tissue provides support to our bodies. So bone, believe it or not, bone is a tissue type and it's connective tissue. Connective tissue transports materials through our body, so the blood and lymph of our body is connective tissue. Connective tissue insulates our bodies, so adipose or fatty tissue is connective, and it protects us. The bones and the fat protect our organs. The cells of connective tissue is widely spaced. Again, in the pictures of microscope slides below, you can see on the left epithelial tissue with the cells tightly packed, and connective tissue in contrast on the right, where the cells colored purple are very far apart from each other. Now let's talk about the composition of connective tissue. Connective tissue is made of two main elements: extracellular matrix, or ECM, and cells. The first element is the extracellular matrix. The extracellular matrix is itself made up of different components. First, the ground substance. This is the fluid or gel or calcified material of the extracellular matrix. It's composed of water and glycosaminoglycans, or GAGs. GAGs are polysaccharides and proteins, also known as proteoglycans. The second component of the extracellular matrix of connective tissue is the fibers. The fibers include collagen. Collagen resists stretching and adds flexibility to our tissues. The second type of fiber is elastin. This adds elasticity to our tissues so they can stretch and recoil back to their original shape. And then finally, the fibers include reticular fibers, which provide support and form a network around the cells of connective tissue. Here you have an electron micrograph of the fibers of connective tissue. The fiber is colored orange, and notice they are thicker, is collagen, and the thinner and blue colored tissue is elastic fibers. The second main element found in connective tissue is the cells. Each type of connective tissue contains its own unique immature class of cells that end in the suffix blast, which means to bud. The blasts of the connective tissue secrete different types of extracellular matrix. For example, fibroblasts are found in loose and dense connective tissue and secrete reticular fibers. The chondroblasts are found in cartilage and secrete cartilage tissue. And finally, osteoblasts are found in bone. Osteo means bone. And they secrete the calcified minerals of bones. Eventually, the cells that create the extracellular matrix mature into a new type of cell. Cells whose names end in blast mature into cells whose names end in the suffix -cyte. These mature cells can't divide as well, and they cannot produce new extracellular matrix, but are there to maintain it. In the picture below, to the left we see fibroblasts. They're creating new fibers in the extracellular matrix. The fibroblasts will mature into cells that we see on the right called fibrocytes, and they are there to just maintain the fibers that are already in place. Now let's talk about muscle tissue. The function of muscle tissue is to contract, to provide movement for the body. The cells of muscle tissue are called myocytes. In fact, the prefix myo means muscle. Muscle is classified by its structure. Muscle tissue can be striated. That means you can see a visible banding in the cells under the microscope. Muscle tissue can also be non-striated, where there's no visible banding in the microscope. Another way to classify muscle tissue is by the way it functions. Some muscle tissue can be controlled consciously and is known as voluntary muscle tissue. Some muscle tissue cannot be controlled consciously and is called involuntary muscle tissue. When you put the different functions and structures of muscle tissue together, we have three types of muscle tissue. Skeletal muscle is muscle tissue that's striated and can be voluntarily or consciously controlled. Think of your biceps. Smooth muscle is non-striated and is controlled involuntarily. Smooth muscle often surrounds the various tubes of our body, like our intestines, our stomach, and our blood vessels. And then finally we have cardiac muscle, which is striated, but is also controlled involuntarily. Cardiac muscle is found exclusively in the heart. Skeletal muscle is found attached to bones. Again, it's striated and can be controlled voluntarily. Here's a slide showing a microscopic picture of skeletal tissue where you can clearly see the striations. Again, smooth muscle is found surrounding the hollow structures of the body. For example, our blood vessels, our lymph vessels, and our intestines are surrounded and controlled by smooth muscle. Smooth muscle is non-striated, and the cells of smooth muscle overlap unevenly. The cells in smooth muscle only contain one nucleus. Contrast that to skeletal muscle, where the cells are multinucleate. In other words, they have many nuclei. Cardiac muscle is found only in the walls of our hearts. Cardiac muscle is controlled involuntarily and is autorhythmic. That means it controls its own beat via pacemaker cells. We can control the rate of our heartbeat by the autonomic nervous system. Finally, our fourth tissue type is nervous tissue. The main functional cell type of nervous tissue is called the neuron or nerve cell. Neurons have three major parts: the cell body, which contains the nucleus and most of the organelles of the cell; the axon, usually it's one long extension or a tentacle-like structure that extends out of the cell. Axons are responsible for sending signals away from the cell. And then finally, the cells have many dendrites. Dendrites are found in multiples, and they receive signals from other cells. The function of nervous tissue is to detect and transmit signals. Nervous tissue responds to different types of stimuli, whether that be pain, pressure, temperature, or light. And nervous tissue sends signals to other cells in our body. Nervous tissue can send signals to other neurons, to muscles, and to glands. Nervous tissue is not composed simply of neurons. It also contains various supportive cells known as neuroglia. Neuroglia support the neurons in the nervous system. For example, neuroglia produce the myelin sheath, or fatty insulating layer. Neuroglia insulate the axons. Some neuroglia are special immune cells that are found exclusively in nervous tissue. And neuroglia can produce the cerebrospinal fluid that protects our brain and spinal cord. And that's it for part 3 of chapter 3.